Okay, so for this class, we're going to take a look at how the breakdown and the synthesis of glycogen can be regulated. So taking a step back, this is a schematic showing the breakdown of glycogen to glucose 1-phosphate and then glucose 6-phosphate on the left. On the right, we have the synthesis of glycogen from glucose 6-phosphate all the way up to glycogen, the individual enzymes and mechanisms for which we talked about last class. In order to regulate the production and the breakdown of glycogen, we want to regulate key enzymes in each step. In the case of a glycogen breakdown, the main point of regulation is glycogen phosphorylase. In the case of glycogen synthesis, our main point of regulation is glycogen synthase. These are the ones highlighted in red, and these are the main sites of regulation of glycogen production and breakdown. And we'll talk a little bit about them in turn. First, we're going to focus on glycogen phosphorylase. If you recall from last class, I asked you to take a look at the regulation of glycogen metabolism in your textbook. We're going to briefly cover that content here. Looking at glycogen phosphorylases, there are several different steps or layers of regulation, as you may recall from your textbook. We're going to start from the top and go all the way down. So at the beginning of a regulatory cascade, we have hormones. These are signaling molecules which circulate your body through your body, signaling your body to do various things. These hormones will then meet with your cells and interact with cellular receptors on the surface of your cell membrane. These cellular receptors stimulate either the production or the destruction of a signaling molecule called cyclic AMP or CAMP. The structure of CAMP is shown over here. This is a common signaling molecule which plays a role in regulating a huge range of cellular processes. These CAMP is generated by an enzyme called adenylate cyclase, which exists at the surface of the cell membrane. And we'll see a little bit about that later. CAMP then goes on to activate protein kinase A, or PKA. Another name for protein kinase A is CAAMP, activated protein kinase, which does kind of reflect its role in this regulatory cascade. We've seen protein kinase A before. In the regulation of glycolysis, protein kinase A can also play a regulation in the role of glycogen synthesis. Protein kinase A in turn upregulates an enzyme called phosphorylase kinase. Phosphorylase kinase then goes on to activate glycogen phosphorylase via phosphorylation, which then in turn goes on to regulate glycogen breakdown. I should point out that both phosphorylase kinase and glycogen phosphorylase are activated by phosphorylation. Conversely, they can be deactivated by protein phosphatase or PP1. This is the same protein phosphatase we saw earlier when we were looking at the regulation of glycolysis. So again, this is an overall scheme showing the regulation of glycogen breakdown all the way from the hormones, all the way down to our glycogen phosphorylase enzyme. Shown over here is an alternate schematic showing this same reaction. Here we start off with our hormones and our cyclic AMP. These go on to then activate our protein kinase, which then activates our phosphorylase kinase and then our glycogen phosphorylase. In this schematic, we show the role of the various kinases in phosphorylating their target enzymes, as well as the role of our phosphoprotein phosphatase 1, or PP1, in inhibiting both glycogen phosphorylase as well as our phosphorylase kinase. An alternate view of this regulatory cascade or regulatory scheme is shown in your textbook, in particular figure 12.26, and that is reproduced up here for you today. Again, we can see our hormones binding to our receptors, which triggers the production of cyclic AMP via our enzyme adenylate cyclase, and then all of its downstream processes from protein kinase A to phosphorylase kinase to glycogen phosphorylase all the way down to glycogen. And again, this is figure 12.26 in your textbook. I will take a minute to focus a little bit on the regulation of glycogen phosphorylase B, because that is a particularly unique system. This regulatory system is unique because it is the activity of glycogen phosphorylase is influenced by its phosphorylation state. As you can see over here in the schematic, glycogen phosphorylase is less active in its dephosphorylated form and significantly more active in its phosphorylated form. However, glycogen phosphorylase can also be regulated or activated by other non-hormonal signals via allosteric regulation. And the interaction between this hormonal regulatory pathway, which is with its various kinases, and its allosteric regulators or inhibitors is rather unique and something which I want to talk about a bit more. So delving into glycogen phosphorylase. Glycogen phosphorylase can occupy two states, either a T state or an R state. The T state is significantly less active, whereas the R state is significantly more active. Shown over here is a schematic illustrating these two. These two states exist in equilibrium. In the dephosphorylated form, i.e. in the absence of our hormonal signaling via pKa and phosphorylase kinase, the T state is favored. However, it can still access the R state, albeit this T state is predominantly favored. When we phosphorylate our glycogen phosphorylase via phosphorylase kinase, we end up with our phosphorylated version. In the phosphorylated form, the R state, or the more active state, is significantly more favored. In other words, in the dephosphorylated form, we have a less active enzyme because the T state is favored. In the phosphorylated form, we have a more active enzyme simply because the R state, or the active state, or active conformation is favored. And conversely, phosphoprotein phosphatase dephosphorylates our enzyme and converts it back into this dephosphorylated, less active form. Where things get unique, where this regulatory pathway is interesting, is the fact that we have an equilibrium. In other words, the phosphorylated form of our enzyme tends to be more active because the equilibrium favors the R state. However, it still can adopt the T state under some conditions. Same with our dephosphorylated state. Our dephosphorylated state is generally less active because it tends to adopt the T state. However, under certain conditions, it can adopt the R state, even in the lack of our phosphorylation. For example, if we look at our dephosphorylated enzyme, significant concentrations of ATP will push it to the R state. Significant concentration of AMP will favor formation of the R state, even though our enzyme is dephosphorylated. In other words, significant concentrations of AMP can push our protein to the active form, even in the absence of phosphorylation and phosphorylase kinase activity. In other words, significant concentration of AMP can overrule the effect of our phosphorylase kinase. The opposite is true. In the phosphorylated or active state, in the phosphorylated or more active state, our enzyme favors the R conformation. However, significant concentration of glucose or glucose 6-phosphate will push it towards the T state. In other words, significant concentration of glucose or glucose 6-phosphate will inhibit our enzyme even in the phosphorylated state. In other words, we can overrule the regulatory effect of our phosphorylase kinase and our phosphorylation state by significant concentrations of our allosteric inhibitors or allosteric activators. And that's an important part of the regulation of glycogen. Yes, glycogen is controlled by its phosphorylation state, but significant concentrations of other allosteric molecules can overrule this to a certain degree. This brings us back to one of the overall themes of metabolism we talked about in the introductory class, about how metabolism is a very distributive process, or the control of metabolism is distributed, right? We have many ways of regulating the activity of this enzyme and the enzyme, either through phosphorylation or allosteric activators and inhibitors, and it is the combined activity of all of these factors which dictates whether our glycogen phosphorylase is active or inactive. In other words, there's no one single point of regulation, but multiple points working sometimes together, sometimes comparatively, to give you a net regulatory signal. These same principles apply to glycogen synthase. I apologize, this should read glycogen synthase at the top. So complex equilibrium of glycogen synthase. So here we have glycogen synthase in its active form or the inactive form. As you can see in the schematic, glycogen synthase is active in the dephosphorylated form. Phosphorylation of glycogen synthase via protein kinase A inactivates it. Dephosphorylation via PP1 activates it. In other words, the opposite of our glycogen phosphorylase we talked about earlier. However, in the phosphorylated form, our glycogen synthase can still adopt the more active R state. Normally, the equilibrium favors the inactive form, as shown by these equilibrium arrows. However, significantly high concentrations of glucose 6-phosphate can overrule the effect of our phosphates and push our glycogen synthase into an active R state despite it being fully phosphorylated. Again, this illustrates the concept of distributive control, and that this particular step in glycogen metabolism is influenced by a wide range of different signals coming from a variety of sources, of which protein kinase A is only one of many. So just for your further interest, there are further readings available in the textbook for you to take a look at, in particular page 406 and 407. This section discusses the specific structural basis for the regulation of both protein kinase A as well as a glycogen phosphorylase. And it's something which you can take a look at later. Finally, we do have several questions which I would like you to take a look at. First, I'd like you to think about the regulation of glycogen metabolism and think about what the advantage is to having such a complex system with so many different allosteric regulators, hormonal regulators, kinase cascade systems, etc., working both together and both in harmony and against each other. And conversely, what is the disadvantage to having such a complex regulatory system? The second question looks at the effect of specific hormones, such as epinephrine, glucagon, and insulin. These are three hormones, not enzymes, which manage glucose levels in the human body. First, I'd like you to think about what the functions are of each hormone. As a hint, you might need to use your textbook at the internet to find this out, but frankly, you can probably punch that into Google. Secondly, based off of the function, can you predict the impact of each hormone on glycolysis and gluconeogenesis, as well as glycogen metabolism? And then finally, predict the impact of epinephrine on cellular cAMP levels and protein kinase A activity.